Let me begin by thanking the conference co-chairs, Drs. Mark Boyd, Nitya Panupak, and Rina Raya for the kind invitation to be part of APACC 2021. Whilst preparing for the talk, I realized that I've been fortunate enough to meet, interact, and come to admire the work of each of them during my many inspiring visits to Bangkok. Here are my disclosures. Whenever I talk about aging with HIV, I always like to begin by stressing that we should remind ourselves that the fact that we nowadays address this topic at all is a testimony of what we've been able to achieve during many years of hard work in developing effective antiretroviral therapies. As a result, people with HIV are increasingly living until older age. Here are the latest data from my own country, the Netherlands, where at the end of 2019, 52% of people with HIV in care were older than 50, up from 11% in 1996. Modeling work by Michaela Smit, shown on the right, projects that this proportion will increase to over 70% by 2030. As I told you, this has everything to do with the success of ART. On a global level, we have made amazing progress where it concerns ART scale-up, with almost 26 million people at the end of 2019 having access to ART. Although this has increased global ART coverage to 2/3 or 67%, at the same time, it also means that as many as 12 million people, or 1/3, do not yet have access, something we cannot and should not accept. Importantly, the figures I just showed you are the global figures. They, however, markedly differ by region as well as between and within countries. For example, as shown on the latest UNAIDS WHO data released last month, for the Southeast Asia region as a whole, highlighted by the red circle, the proportion not covered by ART stands at as high as 40%. And clearly this means we still have miles to go. Nonetheless, with global ART access having increased so much, the numbers of people living with HIV who are over the age of 50 are increasing worldwide. Although the proportion over 50 is higher in high-income countries, as shown by the blue line at the top, than in low- and middle-income countries, shown in the various colors below, in terms of absolute numbers of people, millions more who are over 50 are living in low- and middle-income countries, as illustrated by the orange line. As you all know, aging unfortunately increases our risk of a wide range of comorbid conditions illustrated on the slide. And of course, this is also true for people living with HIV. Again, using data from the Netherlands as an example, one can see that approximately 1 in 5 of those between the ages of 60 and 70 at the end of 2019 had 2 or more comorbidities next to their HIV. Modeling projects that by 2030, it will be the exception for a person with HIV in the Netherlands to be living without any comorbidity. And shown here on the right, the same trend will be moving in a similar direction in a country like Zimbabwe in Africa. In what is now over a decade ago, people started to speculate that comorbidities normally associated with aging might occur more frequently and possibly even at a younger age in people living with HIV, even during the use of effective ART. Central to this hypothesis was that ongoing inflammation, in spite of viremia being suppressed with ART, might be an important contributor, and that this might even result in accelerated and thereby premature aging. In order to address this hypothesis, we were lucky enough to already in 2009 start a prospective cohort study in Amsterdam, the HIV cohort, which a couple of years later also expanded to a European collaboration called COBRA, amongst others with colleagues in the UK who had in the meantime established a similar cohort called POPPY. Although I'll be mainly using examples from HIV and COBRA in the rest of my talk, it is important to stress that fortunately many other cohort studies focused on studying HIV and aging have now been established elsewhere in Europe and around the globe. This slide is by no means meant to be exhaustive, but it shows important examples from each continent. As you know, the demographics of key populations living with HIV markedly differ in settings other than Europe and North America. Think, for example, about the much larger proportion of women living with HIV in Africa. And it's important to have a global portfolio of studies which take these demographic differences into account. Our main aims when we started the HIV cohort were to elucidate whether HIV is causing premature aging. That means whether people experience comorbidities at earlier age, even in a group of people who are treated with effective ART. And if so, why is this happening? Can we identify the risk factors? Can we clarify the pathophysiology? And if we can do so, how can we best intervene in these features? The key features of the cohort was to recruit an appropriately matched control group of participants who, with a similar distribution in demographic characteristics such as sex and age, behavioral and lifestyle factors such as the proportion being MSM, smoking, using recreational drugs, experiencing STIs or viral co-infections. For the HIV positive patients, we selected a group that were virtually all on combination therapy with suppressed viremia, thereby reflecting the contemporary HIV positive population reaching an older age in countries with widespread access to combination therapy. And finally, we invested in performing identical and detailed data collection of both outcomes and potential confounders through standardized procedures and protocols in both groups, which allowed us to, of course, appropriately adjust for such confounders in our analyses. A key point to keep in mind is that recruitment of appropriately matched controls and appropriate adjustment for potential confounders are really of vital importance for studying aging and age-associated comorbidities in the context of HIV, if the question is that you're asking is to study the effect of HIV and ART over and above the effect of other relevant demographic and lifestyle characteristics. Concerning the question if comorbidities associated with aging occur more often and at younger age in people living with HIV, many studies, including our own, as shown on the slide, generally have found a wide range of comorbidities to occur more frequently in people living with HIV, including in those with sustained suppressed viremia on ART. An important comment to make is that studies have differed in the size of the differences observed between those with HIV and HIV-negative controls, and also in the extent to which an increased comorbidity risk was independently associated with being HIV positive. In many instances, the discrepancies between studies can be explained by the markedly different demographic makeup of the controls and/or the inability to adequately control for important known traditional risk factors for these comorbidities. For example, because they were not known or not assessed in the controls. And in my view, this is a key issue where our own study has been able to make an important contribution. I don't want to spend too much time on discussing the underlying drivers of increased comorbidity risk in people living with HIV, including the important role of inflammation, as this could easily justify a separate talk of its own. The key main point I'd like to make, though, using this slide, which references the results of several published analyses of our HIV and COBRA studies, is that the higher comorbidity risk in people living with HIV in case of each specific comorbidity was multifactorial. Lifestyle-related factors cardiovascular disease risk factors, body composition, co-infections such as HCV, immune activation, inflammation, use of certain ARVs, as well as markers of severe prior HIV disease, all potentially contributed, but to a different extent. In other words, the risk factor pattern was not generalizable for all comorbidities, whereas some risk factors were associated with multiple different comorbidities, others clearly were not. And this shows that when performing such studies, although there may be value in lumping comorbidities together, studying each of them individually is definitely also required. What I've told you so far are all results of cross-sectional studies. But what happens over time in people living with HIV with sustained viral suppression on treatment? Does their risk of developing comorbidities accelerate compared to in HIV-negative individuals? The answer, like we've seen for the cross-sectional studies, seems to be that not every comorbidity behaves the same. For example, when looking at different MRI measures of brain structure as well as a formal neuropsychological assessment, we found that the differences observed in these measures at the time of entry into the COBRA study 2 years later had remained the same between HIV-positive and negative participants, as illustrated by the lines not showing a greater diversion at the 2-year follow-up assessment. In contrast, when looking at changes in spirometry over a period of 6 years, whilst accounting for the effects of smoking, both 1-second forced expiratory volume and forced vital capacity declined significantly faster in HIV-positive participants in the HIV cohort, with a similar trend in the smaller subset of participants who had never smoked. This suggests that different from what we had observed for the brain, HIV may in some way continue to accelerate lung tissue damage in spite of viremia being suppressed. Now let's turn briefly to the issue whether HIV may affect the actual process of aging. Let me start by showing you our results of measures of immune senescence in the COBRA cohort, as these very nicely illustrate the importance of how the same finding may be interpreted quite differently depending on which control group one uses. When comparing our HIV-positive participants in red to a control group of age-matched blood donors in gray, HIV positives showed more immune senescence, both in the CD4 cell and CD8 cell compartment, than the blood donors. But no difference was observed compared to our matched HIV-negative controls. Had we just used healthy blood donors as controls, we would likely have concluded that HIV by itself is associated with immune senescence, whereas using the appropriately matched HIV-negative controls, the conclusion is that this isn't the case. In other words, be aware who you compare. Interestingly, our finding that at least for this particular biomarker, HIV-negative participants also differed from healthy blood donors suggested that factors other than HIV must also be playing a role in driving a process like immune senescence, and possibly also this could be the case for other aspects of aging. We tried to get more insight into what such other factors might be by performing an analysis which was focused solely on the HIV-negative men in the HIV cohort. And this showed that HIV-negative MSM had lower CD4-CD8 ratios, largely driven by higher CD8 counts, than men having sex with women, with the most marked difference and effect observed in MSM with a higher number of recent sex partners. And we found that this could at least in part be explained by a higher seropositivity rate for cytomegalovirus in those MSM engaging in higher risk sexual practices. Using a measure of biological age based on a composite of 10 biomarkers identified by the EU-funded MarkAge project, which was aimed at identifying the best marker of biological age, we found that people living with HIV on suppressive ART in red had a greater degree of biological age advancement than our HIV-negative controls in blue in the COBRA cohort. But, similar to what I showed you for the markers of immune senescence, both groups, HIV-positive and HIV-negative controls, showed age advancement compared to the healthy blood donors in green. Similarly, using methodology on the basis of MRI imaging of the brain to assess premature brain aging, a method originally developed by our colleagues in the UK in the context of studying premature brain aging in the brains of victims of traumatic brain injury, we found a greater than expected mean brain predicted age in our HIV-positive in red than HIV-negative participants in blue in this cross-sectional baseline analysis from COBRA. Importantly, similar to what I have shown you earlier for structural brain changes on MRI and neuropsychological assessment, over a 2-year follow-up period, the observed difference in brain predicted age between the two groups at baseline remained the same 2 years later. In other words, there was no evidence of accelerated brain aging in those with HIV, at least not over this limited period of observation, and importantly remind yourselves this is a population well suppressed on ART. So, what are the practical implications of everything I've told you so far? I think the clinical management of individuals with HIV as they age needs to reflect the multifactorial nature of comorbidity pathogenesis. Apart from striving for early HIV diagnosis and the use of least toxic and most effective ART to ensure sustained viral suppression, there currently really are no proven adjunctive treatments to directly target, for instance, residual inflammation or the aging process itself, although studies in this area and trials are ongoing. So we're left by the fact that it's key to stimulate behaviors aimed at a healthy lifestyle, especially concerning substance use, exercise, and nutrition. Interventions to prevent and manage specific comorbidities are largely based on general population guidelines, with several very useful internet-based tools being available, some shown on the left of the slide, to help guide healthcare providers with respect to important considerations in the context of HIV. So, to summarize the main points of my talk in terms of conclusions and lessons learned. The number of people living with HIV reaching older age will continue to grow globally. Comorbidities associated with aging are generally more prevalent in people living with HIV, but the degree to which they are independently associated with treated HIV differs, both for specific comorbidities and between studies. Proper interpretation of study results with respect to the contribution from HIV is very much dependent on the comparability of the HIV-negative controls. Higher comorbidity risk in people living with HIV is clearly multifactorial, but risk factors are not necessarily uniform for every comorbidity. Accelerated organ dysfunction in people living with HIV with sustained viral suppression on ART may occur, think of the pulmonary data I showed you, but it's not uniform across organ systems, think of the brain data I showed you. There may be some degree of sustained accentuated aging, but little evidence, at least from our data, of it being truly accelerated in people living with HIV with sustained viral suppression on ART. And finally, the clinical management needs to reflect these insights and needs to be made accessible for all. Let me also take this opportunity for those watching with a special interest in this area of research to the upcoming next edition of the International Workshop, which has now been renamed the International Workshop on Long-Term Complications of HIV as well as of SARS-CoV-2. It will take place online during the week of December 9. More information can be found at the website on the bottom of the slide, and I want to stress that we especially welcome young investigators from around the world to submit their abstracts, including from your part of the world, of course. Finally, I would like to acknowledge all our HIV and COBRA study participants, our many collaborators, our funders, and the many colleagues I've had the pleasure to interact with over the years, and I want to thank you for listening.